Welcome to the PubMed Daily Review for April 8th, 2026. Today we scanned 2,117 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Vasculitis and Autoinflammatory Syndromes. These papers explore inflammatory conditions affecting blood vessels and the immune system, including autoimmune and genetic disorders. This cluster covers 489 papers. Representative work includes eosinophilic granulomatous polyangiitis emerging after omalizumab administration. An asthma patient without systemic corticosteroids develops mononeuritis multiplex after 22 days, Rowell syndrome with lupus podocytopathy, a diagnostic challenge, and regression of a refractory pulmonary lesion in granulomatosis with polyangiitis following the addition of avacopan. Trend 2 of 5, Biomedical Research and Disease Modeling. Investigating biological mechanisms and disease processes using diverse experimental and computational approaches. This cluster covers 527 papers. Representative work includes Iratum, advanced microfluidic and 3D cell culture platforms for modeling vascularization in diabetic foot ulcers, a systematic review of translational challenges and perspectives, and the RNA helicase F4A as a novel target in insect cells to combat arboviral infections. Trend 3 of 5, Cancer Biology and Treatment. This trend explores diverse cancer types, treatments, patient outcomes, and survivorship challenges. This cluster covers 261 papers. Representative work includes dual suppression of stemness and redox adaptation in glioblastoma through filaggrin upregulation by an abiraterone-based HBsAg inhibitor, basal ganglia glioblastoma with lung adenocarcinoma tumor-to-tumor metastasis, and atypical early imaging presentation, illustrative case, and long-term control of grade 3 intracranial solitary fibrous tumor achieved with pazopanib and stereotactic radiotherapy, illustrative case. Trend 4 of 5, Patient Education and Health Technology. This trend explores how technology enhances patient understanding and management of health conditions. This cluster covers 564 papers. Representative work includes disease course of axial spondyloarthritis in pregnancy, the relationship between joint health, kinesiophobia, physical activity level, and functional capacity in hemophilia patients, a case control study, and quick response codes in the breakfast cereal aisle. Prevalence, landing page, and product characteristics. Trend 5 of 5, Neurological Disorders and Case Studies. These papers explore neurological conditions, their impacts, and diagnostic approaches through case studies and analytical methods. This cluster covers 276 papers. Representative work includes psychological correlates of pain, catastrophizing, and disability in patients with Achilles tendinopathy, the roles of resilience and psychological inflexibility, neurosarcoidosis mimicking multiple meningiomas, illustrative case, and letters to the editor, radiological, parasitological, and biological considerations regarding alleged adult tinea solium brain infection. Up next, our first paper out of 5, Overcoming Immunotolerance in Chronic Hepatitis B, Efficacy of Granulocyte Macrophage Colony-Stimulating Factor-Based Immunotherapy in Achieving Hepatitis B Surface Antigen Seroclearance, by Shuanggang and Bin Wang from Key Laboratory of Medical Molecular Virology, MOE/NHC/CAMS, College of Basic Medical Sciences, Fudan University, Shanghai, China. Let's break down its key contributions. Chronic hepatitis B is a major global health problem affecting millions worldwide. Despite available treatments, a functional cure, defined by the clearance of the hepatitis B surface antigen, HBsAg, remains elusive for most patients. This is largely due to the virus's ability to induce immune tolerance, preventing the body's own defenses from effectively clearing the infection. This study explores a new immunotherapy approach to break that tolerance. This study enrolled 72 patients with chronic hepatitis B and divided them into 3 groups. The first group received standard nucleotide analog therapy, ADV, alone. The second group received ADV plus interferon alpha. The third experimental group received ADV, interferon alpha, and a novel immunotherapy regimen called TCHR-LGM-VAC, which involves granulocyte macrophage colony-stimulating factor and an HBV vaccine. The primary goal was to see if this new regimen could lead to the clearance of HBsAg. The results were promising. While the control groups saw minimal HBsAg clearance, 8.7% of patients in the THR-LGM-VAC group achieved HBsAg seroclearance. Furthermore, over 21% in this group experienced a significant reduction of more than 2 log 10 in HBsAg levels. This was associated with a notable decrease in regulatory T cells, which promote immune tolerance, and an increase in beneficial anti-HBV T cell responses. The treatment was generally well tolerated, with only a slight, temporary rise in liver enzymes observed. This study suggests that the THR-LGM-VAC immunotherapy offers a new avenue for achieving functional cure in chronic hepatitis B by effectively breaking immune tolerance. The findings are particularly encouraging for patients with lower baseline HBsAg levels. While these results are exciting, further studies are warranted to refine the treatment protocol and identify the optimal patient populations to maximize the benefits of this innovative approach. Up next, our second paper out of 5, a stroke organoids-multiomics platform to study injury mechanism and drug response by Wendy Zhu and Ling Lung from A Stem Cell and Regenerative Medicine Lab, Institute of Clinical Medicine, State Key Laboratory of Complex Severe and Rare Diseases, Translational Medicine Center, Peking Union Medical College Hospital, Chinese Academy of Medical Sciences, and Peking Union Medical College, Beijing, 100730, China. Let's break down its key contributions. Stroke remains a devastating global health crisis with limited effective treatments. A major hurdle is the lack of human-specific models to accurately study strokes, complex mechanisms, and test potential therapies. This research introduces a novel approach using human brain organoids to bridge this gap. To tackle this challenge, the researchers developed a sophisticated platform. They created human brain organoids from stem cells and induced a hypoxic stroke model. This was combined with advanced multi-omics techniques, including proteomics and single-cell RNA sequencing, alongside traditional histology, to deeply analyze the cellular and molecular changes occurring during stroke. The results were compelling. They identified 3 distinct types of astrocytes that responded differently to the hypoxic environment, shedding light on their role in stroke-induced brain tissue changes. Furthermore, cortical neurons showed clear signs of apoptosis and aging following hypoxia. Importantly, the Dengzhang capsule demonstrated a potential therapeutic role by influencing lipid metabolism-related pathways. This research represents a significant step forward. The combination of brain organoids and multiomics provides a promising new strategy for studying stroke pathogenesis and offers a valuable platform for future drug screening. While these findings are exciting, further research is needed to translate these discoveries into effective clinical treatments for stroke patients. Up next, our third paper out of 5, dysfunctional lipid-induced secretion of glucagon like peptide 2, GLP-2, but not of incretins glucagon like peptide 1, GLP-1, / glucose-dependent insulinotropic polypeptide, GP, promotes metabolic dysfunction, associated steatotic liver disease, MASLD, onset and progression through gut barrier disruption and endotoxemia by Giovanna Pietrangeli and RG from Indina SBA. Let's break down its key contributions. Metabolic dysfunction-associated steatotic liver disease, or MASLD, is a growing concern, encompassing a spectrum from simple fatty liver to more severe MASH. While we know fat plays a role, how our bodies handle fat after we eat and how that impacts the liver is still a puzzle. This study dives into that dynamic post-meal response to uncover new insights into MASLD. To investigate this, researchers studied 2 groups. First, a cross-sectional group compared patients with MASLD, MASL, and healthy controls after a standardized fat meal. They measured key hormones like GLP-2, gut barrier markers, and signs of inflammation. Then, in an interventional study, MASH patients were treated with a curcumin supplement or placebo for 72 weeks, with similar tests performed before and after. The study found a clear pattern. As liver disease worsened, the body's ability to secrete GLP-2 after a fat meal significantly decreased. Simultaneously, gut barrier integrity weakened, leading to increased endotoxemia. Importantly, a curcumin supplement improved GLP-2 response and gut health in MASH patients. Lower GLP-2 levels were strongly associated with the presence and severity of MASLD and even predicted MASH resolution. This research highlights that the way our gut and liver handle fat post-meal is crucial in MASLD. The findings suggest that targeting GLP-2 secretion, and improving gut barrier function could be a promising new strategy for treating MASLD. This approach might offer unique benefits, particularly for lean individuals with MASLD who don't typically qualify for weight loss-focused therapies. Up next, our fourth paper out of 5, Unraveling Genome-Wide Mosaic Microsatellite Mutations at Single-Cell Resolution by Chunyi Wang and Yimei Do. Let's break down its key contributions. Microsatellites, also known as short tandem repeats, are fascinating parts of our genome. They're highly mutable and play roles in how our genes are regulated, and they've even been linked to various human diseases. However, spotting these tiny changes, especially when they occur in a mosaic pattern within individual cells, has been a major hurdle. This is due to a combination of tricky lab techniques and the inherent complexity of these mutations. To tackle this challenge, the researchers developed a powerful new tool called BaseMon-STR. This algorithm is designed to accurately detect mosaic STR mutations at the single-cell level. They applied this tool to analyze various human tissues, including brain cells, blood cells, and lung cells, comparing younger and older samples. Their approach allowed them to uncover a previously hidden landscape of genetic variation. The results were quite striking. They found that mosaic STR insertions and deletions accumulate in aging cells, both those that divide and those that don't. Notably, neurons in the prefrontal cortex showed a significantly higher number of these mutations compared to B cells or lung cells. This increase was even more pronounced in older neurons, with a notable rise in longer STR deletions. These mutations were also found to be concentrated near the start sites of genes and active regulatory regions. This work is significant because it provides a new way to discover disease-related mosaic STR mutations across the entire genome. It opens up a whole new area of research into how these mutations change as we develop and age. The findings hint that these dynamic genetic changes, especially in neurons, might play a role in age-related brain function and diseases, paving the way for future investigations. Up next, our fifth paper out of 5, Intraperitoneal CF33-hNIS combined with PD-L1 blockade eradicates gastric cancer peritoneal metastases and prevents recurrence via durable T-cell memory by Annie Yang and Yang-Hee Wu from Department of Surgery, City of Hope National Medical Center, Duarte, California, USA. Let's break down its key contributions. Gastric cancer is a major global health challenge, and when it spreads to the peritoneum, the prognosis is grim. Current treatments are often ineffective against this widespread disease. We need new strategies to target these peritoneal metastases. Oncolytic viruses like the engineered CF33-hNIS offer a potential new approach by directly killing cancer cells and stimulating the immune system. To test this new therapy, researchers used a mouse model that mimics gastric cancer spreading to the peritoneum. They administered an oncolytic virus called CF33-hNIS directly into the abdominal cavity, either alone or in combination with an immune-boosting drug that blocks PD-L1. They then analyzed tumor growth, immune cell activity, and whether the treatment could prevent the cancer from coming back. The results were very promising. The combination therapy, specifically a high dose of CF33-hNIS given intraperitoneally along with anti-PD-L1, led to complete tumor regression in 75% of the mice. Even more exciting, mice that achieved complete regression were protected from a later tumor challenge, indicating the development of lasting immune memory. This suggests the treatment not only eliminated the existing tumors, but also prepared the immune system to fight off future recurrences. This study demonstrates a powerful new approach to treating gastric cancer that has spread to the peritoneum. By delivering the oncolytic virus directly into the abdominal cavity alongside an immune checkpoint inhibitor, researchers achieved remarkable tumor eradication and established long-term immune memory. This preclinical success provides a strong foundation for moving this innovative therapy into human clinical trials, offering hope for patients with this challenging disease.